Now that I'm fueled up, here we go. So as I mentioned today, we have our Introduction to Animals PowerPoint. We're going to go through basic animal characteristics. We're going to spend the rest of the time talking about differences between animals. Today we get to talk about similarities between the groups. And then we'll talk about different body plans as we make our way into animal diversity. We already have our first phylogenetic tree on the far side of the board that kind of maps out some of this diversity. We're going to be breaking it into more detail in Wednesday's lecture. Okay, I said the first PowerPoint, we're going to go through characteristics that all animals have in common before we start separating the different groups from one another. All animals we're going to see are multicellular eukaryotes. They're ingestive heterotrophs as well. Maybe the first thing is worth breaking apart, multicellular eukaryotes. We've seen multicellular eukaryotes before, but they had cell walls, things like fungus and things like plants. These are multicellular eukaryotes. We're going to see that they lack cell walls, but instead of a cell wall, they have this extracellular matrix. It's going to be a substance that it's secreted by cells and kind of fills the space in between cells. If this was an anatomy and physiology class, we would be talking about different types of tissue, and you have different types of tissue depending on the different types of extracellular matrix. This is a fluid connective tissue in blood, and the extracellular matrix we call plasma. It's mostly water. It's like 90% water. A very fluid extracellular matrix, where this hyaline cartilage is a much more gelatinous extracellular matrix. It gives this group of cells a little bit of resiliency or strength. So lack cell walls, they have an extracellular matrix. To jump back to this slide, we said like plants and like fungus, they're multicellular eukaryotes. But unlike plants, they're heterotrophs. And unlike fungus, their digestive enzymes are on the inside. They're not absorptive heterotrophs. When I take this coffee, I don't just dump it on my head and let the caffeine and sugar be absorbed by my body like I would if I was a fungus. I have to ingest it and let it contact my digestive enzymes on the inside. So ingestive heterotrophs lack cell walls, have that extracellular matrix, and then the last characteristic is that at some point in their life cycle, they're going to have mobility. We have this mobility at all points in our life cycle, but we're going to see some organisms, like the ones that we'll see next time, these cnidarians, they can alternate between different parts of their life cycle, where some parts of their life cycle, they grow kind of like a branching tree, where they're attached to the substrate, they're attached to the ocean floor, and they look like a tree. This is what we're going to call a polyp form of a cnidarian, where some of these branches are going to produce these little feeding structures where they reach out and grab onto food particles. And other structures are going to start to pop off these little jellyfish, these little medusa buds. So at least some part of the life cycle has motion or mobility. Other parts of the life cycle might be fixed to some type of substrate. Here's another slide that shows us something that is obvious to us. I mean, we're familiar starting off as a single zygote that was one diploid cell and grew to a bunch of diploid cells into our adult form. It was mitosis that gave us identical cells. And again, this is nothing new to us, but the haploid stage of our life cycle, there is just a single-celled haploid stage of our life cycle. There's no multicellular haploid structure. In plants, we saw that alternation of multicellular diploid, multicellular haploid. If we go all the way back to protist, if we think about things like red algae, brown algae, green algae, those were multicellular, but all of those cells were haploid cells. There wasn't a multicellular diploid structure. When they formed a diploid structure, it was a single-celled diploid structure that waited those harsh conditions out and then broke back into haploid cells. Animals have a multicellular diploid phase, but no multicellular haploid phase. Here's what we need to add in terms of animal growth and development. We see that all animals are going to start out as a single diploid cell. We call that the zygote. But this process of going from one single diploid cell to a handful of diploid cells that are all identical, this process of cell, let me say this carefully, this is a process of cell division without any cell growth. Notice how the cells don't get any bigger. The cells aren't growing. The cells are also not differentiating. This cell division without cell growth or cell differentiation is what we call cleavage, the second word here on our PowerPoint. So we know our zygote, single diploid cell. This process that takes us from one diploid cell to a bunch of identical diploid cells is cleavage. At the end of this cleavage process, we're left with a 64-celled hollow sphere. All of those cells are genetically identical, and they have not yet committed to being any particular type of tissue. Any one of those cells contains all the genetic information that they could develop into nervous tissue, muscle tissue, epithelial tissue, connective tissue. These are what we call true stem cells. But in this hollow sphere of identical cells, we call a blastula. So we're going down the list. We've got our zygote. The process that takes us from a zygote to a blastula is the process of cleavage. We get cell division without any cell differentiation. Cells have not yet started to specialize. But that changes after we pass this blastula stage. How we know that we're looking at a gastrula is that a gastrula, we start to see cells congregate at one end of this sphere. You can start to see some of these cells bunching up. It's kind of starting to get lopsided. And in this picture, let me kill this other light just for a second. Maybe that's a little bit easier. We can see that little indentation of the cell. That's a layer of tissues that is starting to make its way towards the inside of the cell. This is where we start to get two layers of cells. An inner layer of cells. Let's list that. So we have this outer layer of cells that remains on the very outside perimeter of this growing embryo. That's going to be our ectoderm. And then what we can see starting to make its way towards the inside of the cell, this tube that is starting to elongate, is going to be the inner layer of cells that we call the endoderm. We're going to see animals with a tissue layer are going to have either two tissue layers. Some animals are going to add a third layer, this mesoderm. So we need to be aware of three possible tissue layers or germ layers. We'll talk about what tissues or what organs form from these three germ layers in just a second. And even some animals will modify those organs as they go from their larval stage to their adult stage. Think like butterflies or frogs, for example. Here's this is a sketch of the same process. We go from a single diploid cell to a handful of identical diploid cells by way of cleavage, cell division without cell differentiation or growth. After this blastula stage, we've got a hollow sphere of cells, no cell differentiation yet, but that changes as soon as we get to this gastrula phase. We start to see an endoderm layer of cells distinguish itself from the outer layer, this ectoderm layer of cells. And then a side note that we want to just plug for later, this first little opening that is created whenever the endoderm layer of cells kind of folds in on itself, that first little opening is called a blastopore. And we're going to see in one group of organisms towards the bottom here, that blastopore is going to develop into the mouth, where another group, the deuterostomes that we belong to, that blastopore we're going to see actually ends up developing into the anus. So fate of blastopore is going to be one of these characteristics that we use to distinguish animal groups from one another. I tried to write some notes on the board in our phylogenetic tree. I'll explain those as we go through. I can write them bigger if needed. What we can see here is one of these growing embryos that has three germ layers, and there's something that I want to connect with the number of germ layers. Maybe I'll try this just saying it with words. I feel like I have to write everything down, but I probably don't. Animals that have just two germ layers, just the ectoderm and the endoderm, we're going to see these animals, we're going to call them diploblastic, just two layers. And kind of the side effect of having only two layers of cells is that these organisms are going to be radially symmetrical, like a jellyfish. They they encounter the environment the same at all sides of the organism. There's no end of the organism where all of the nervous tissue is congregated. Whereas animals that have 3 germ layers that add this mesoderm, we can see the additional mesoderm in red. Knowing that an animal has 3 germ layers, we automatically can tell something about the body plan of this organism. All triploblastic organisms with 3 germ layers are going to be bilaterally symmetrical. So if you've got all 3 of these germ layers, you're going to be like us, where you could have a mirror image of either side of us, bilaterally symmetrical. The other notable thing about having 3 germ layers is it's these organisms that have cephalization, this formation of a head, where all of the nervous tissue starts to congregate at one end, because that's the end that encounters the environment. As these 3 germ layers continue to progress, what we start to see are different organ systems develop from different germ layers. And this outermost layer, this ectoderm that we're seeing, from this ectoderm you start to get neural tissue that forms. In fact, if we want to list some organ systems and which germ layers they derived from, here's a good slide to maybe put a star next to. For the ectoderm, the outer layer of cells, some common organs that form from this is all of the epidermis, your integument. The skin develops from this ectoderm layer of cells, as well as the nervous tissue. You've got a lot of nerve endings in your hand and on the surface of the body. So ectoderm, think the skin and the nervous tissue. When we get to the mesoderm, that middle layer of the body, some of the common organ systems that come from this mesoderm, think the skeletal system, so your bones and your muscles, the muscle system, as well as blood vessels. The blood vessels are lined with muscles themselves that can constrict or dilate. So all of this muscle tissue, all of the skeletal system derived from that mesoderm. And then we get our innermost layer of cells. So think these are the deepest layer of cells. They're going to be the ones that line the digestive tract. So we would include the digestive tract and any of these accessory organs like the liver and the pancreas, some of those main accessory structures to the digestive system. So the innermost organ systems are derived from this endoderm. I mentioned in addition to having 3 germ layers, these organisms are going to have cephalization. They're going to have one end of the organism where all of the nervous tissue starts to congregate, and that's the end that encounters the environment. Some organisms are going to modify their body plan significantly as they go from their larval stage to their adult stage. We are not part of this group, so our body plan stays roughly the same from our infant stage to our adult stage. We just grow a little bit larger, whereas other body plans can change quite drastically from their larval stage to their adult stage. A tadpole or a butterfly would also be a good example of this, but a tadpole works where the larval stage doesn't have any legs at all. It breathes through these gills. It has a tail that it swims with. And then before it hits its adult stage, it's, what's interesting is that if we look inside the cells, the cells inside the tadpole, if we go inside the nucleus of those cells, they've got all of the genetic material to build legs, to build the lungs that they need to breathe air and absorb oxygen through their skin. It's just that those genes aren't turned on until they hit a certain stage in development. Our own reproductive organs are the same way. They aren't activated until we hit a certain stage in development. And then when certain genes turn on, it can lead to modification of certain organ systems. So some organisms do this more pronouncedly than us. Frogs and butterflies would be drastic examples of morphogenesis, so changing their body plans up. Thinking about body plans, we're going to separate. I'm going to turn this light on again real quick so we can see the stuff that's on the board. When we break animals up into their separate groups, I'm just going to visit the board because this is a nice chart. It's also a slide that you have. It's one of the last slides in the PowerPoint. But it shows us this group of animals that I've actually included an outgroup here. These choanoflagellates, they're protists. In fact, colonial protists, unlike animals that are multicellular, we said colonial organisms, there was more than one cell, but there was no cell differentiation. All of those cells are doing the same thing. In animals, we're going to see cell differentiation. So animals are multicellular, but like we're about to see, when we look more closely at the animal group, not all of them are going to have true tissues. So the group of what we call true animals or eumetazoa, these are true animals that have some type of body tissue. There is a group, the sponges, that are considered animals because they're multicellular, but they don't have true tissues. We're going to learn that if you don't have tissues at all, you also don't have any symmetry. So parazoa, these mean animal adjacent. They're multicellular, like we said, but they don't have an endoderm and an ectoderm. So they don't have any symmetry. It looks like I don't have a picture of one, but I did order off of Amazon a whole collection of sponges that should be here tomorrow. In fact, I just got an email saying that they will be. So by the time we get in here on Wednesday, I should have a station set up over there that show us different types of sponges. And our sponge example are going to be all of these organisms that lack symmetry, they lack tissues. Whereas the next group that we see, these cnidarians, those are going to be the first group of organisms that we're going to classify and look at that belong to this group of eumetazoa. So of this entire group of eumetazoa, we're going to see only the cnidarians and this other group called Ctenophora. These are the comb jellies. All of the jellyfish fit in this cnidarian group, and Ctenophora, these are comb jellyfish. They're all radially symmetrical. They have just two germ layers, that ectoderm and the endoderm. But at least they have germ layers, so that puts them in the group of true animals. If they had three germ layers, they're going to be triploblastic. They're also going to be bilaterally symmetrical. So we've got a whole group of bilaterally symmetrical organisms that we'll spend a few weeks looking at. We get to see platyhelminthes as our example of triploblastic organisms in the next lab. But let's look at this slide that shows us radially symmetrical organisms, kind of like this flower pot. You could divide in multiple ways to make mirror images of this organism. Another way of thinking about it is that radially symmetrical organisms, they encounter the environment the same at all sides of this organism. So there's no place for the nerve tissue to congregate at one end. There's no cephalization. Whereas bilaterally symmetrical organisms do have this congregation of nerve tissue at one end of the organism. So this is our head region. Cephalization is something that we see in only bilaterally symmetrical organisms. So we see the mouth parts, the antenna, all the little pinchers congregate at just the head end of this organism. Looking at this picture, these are all sea anemones. We can see radial symmetry, so that tells us just two germ layers, and we know automatically they fit in this diploblastic group of organisms, either cnidarians or Ctenophora. And when we look at cnidarians, they do this, like I had erased, well, I've erased it now, but I had up here a second ago that diagram of the jellyfish life cycle, where sometimes some part of their life cycle is sedentary. It's attached to the bottom of the ocean floor, and it grows more in what looks like a tree, where some of those branches of the tree are feeding off nutrients suspended in the water column, and other branches are putting off these jellyfish, the free swimming part of the life cycle, whose job it is to go and reproduce. Drop those fertilized zygotes elsewhere so the organism can spread. So diploblastic organisms, that's the term that we will use to describe two germ layers, and you know if it's got two germ layers, it's radially symmetrical. Just like if it has three germ layers, if it's triploblastic, we don't even need to see the organism. We know whatever it is, it's going to be bilaterally symmetrical. The big advantage with bilateral symmetry, like we said, was cephalization. All that nervous tissue could congregate at one end of the organism. And we've got a couple of models that we'll see next time. And one of them is this, well, we've got two models. One of them is of this sea anemone. It's kind of a model of that picture that we just looked at, where we can see an organism that's radially symmetrical. When you flip this thing around, we don't see any cephalization. versus this flatworm where we've got two different layers of the same flatworm. We can see cephalization, a clear head region, and you start to see some of the nervous tissue congregate at one end. It even has two simple eye spots. So cephalization, triploblastic. Let's see what else we can list. Diploblastic, like we just said, two germ layers. We know these are radially symmetrical organisms. Triploblastic, meaning three germ layers. These organisms are bilaterally symmetrical. So if you see something like this and you're asked whether it's radially symmetrical or bilaterally symmetrical, without even seeing how it grows up, we can recognize mesoderm. We know it has to be bilaterally symmetrical, triploblastic. Okay. Doing okay? Am I going too fast? Going all right? On to body cavities. We need to explain what a body cavity is before we can talk about which organisms have it and which don't. And going back to our chart over here, so far we're thinking about what organisms are multicellular and which ones are not. So if you're multicellular, you're part of this animal group. Then within the animal groups, do you have tissues or don't you have tissues? If you do, then you're in this eumetazoa group, so we've excluded the sponges. Well, if you have tissues, is it two germ layers or three germ layers? If it's just two germ layers, you're radially symmetrical, you belong to this branch. But if we're triploblastic, we're going to be bilaterally symmetrical, so we need to look for some other thing that's going to distinguish bilaterally symmetrical organisms. So we ask the question, does it have a true coelom or does it lack a coelom altogether? So deuterostomes and this other branch that was formerly called proterostomes, they have either a true or a false coelom, whereas our flatworms, or acoelomorphs, as their name is telling you, they don't have any coelom whatsoever. So what is a coelom? It's going to be basically a cavity. It's a chamber found within the mesoderm. So for it to count as a true body cavity, as a true coelom, it has to be in the middle of this mesoderm. Let me show you what I mean. This is a flatworm. It lacks any kind of coelom whatsoever. So there's our ectoderm in blue, our endoderm in yellow, and our mesoderm in red. For this to be a true coelom or a true body cavity, that has to be in the middle of this red part, in the middle of the mesoderm. Don't confuse a body cavity from a digestive tube. So if this flatworm were to eat a piece of gum or something, it could pass through this digestive tube in the middle of that endoderm. That's not the body cavity. So digestive tract, not a body cavity. A body cavity is this hollow space in the middle of the mesoderm. It allows these digestive organs to move independently of the body wall. So our intestines can move around and fold over. That's because they're inside this actual body cavity. Allows space for folding. It helps protect and cushion those delicate internal organs. So there are advantages to having a body cavity. This animal does not have a body cavity whatsoever. There's no space in the middle of that mesoderm. This animal does have a body cavity, but it's not considered a true body cavity because it's not within the middle of the mesoderm. This body cavity is in between the mesoderm and the endoderm. So this is what we call a false body cavity or a pseudocoelomate. These roundworms, these are parasitic worms that are found in pigs often. These are pseudocoelomates. They have this body cavity between endoderm and ectoderm. So not a true coelom. This is a picture of a true coelom where there is a body cavity in the middle of the mesoderm. We have a body cavity like this. We can see the earthworm also has a true coelom, just meaning that its digestive organs and everything are within this true body cavity, within the coelom, the pocket in the middle of the mesoderm. Okay, so acoelomate, our flatworms are an example of this. Pseudocoelomate, roundworms are an example of this. And then annelids, our segmented worms, or even us, are an example of coelomates. So we wanted to separate not having a coelom whatsoever, like the flatworms, from having either a true or a false coelom, which puts you into this category of deuterostomes, or what was formerly known as proterostomes. You can see this term proterostome is on the board. You can think of that term proterostome as describing these last two groups, Ecdysozoa and Lophotrochozoans. These are weird terms that we'll describe, but they are formally known as the group Proterostomes. So now we're looking at this last branch, and we want to separate Deuterostomes from this group that was formerly called Proterostomes. The differences between these two branches is that the fate of that blastopore is different. In the Deuterostomes, it becomes the anus. In the other two groups, it becomes the mouth. And then we'll see in a picture that the type of cleavage is different between Deuterostomes and then the last two group, Ecdysozoans and Lophotrochozoans. Let's think about that real quick. We mentioned earlier in our lecture what cleavage is. What do you think of when, like if you were to describe the process of cleavage to somebody when you walked out of here, how did we define that? I chopped up my words when I was trying to say it, but I'm sure you can do better than I did. I'm hearing something, but I'll say it louder to the back of the room. What? I'm hearing some good stuff. It's coming.
It's like an indent in between cells.
There we go. Yeah. So I think. So he's saying splitting without increasing in size. So how I was saying that, and he's saying it the same way with different words, is that you have cell division without cell growth or without cell differentiation. So the cells aren't starting to specialize. They're not starting to grow. They're just dividing in number. So cleavage is cell division without cell differentiation. And then, Isabella, I think you were about to say the other part, which is this difference between spiral versus radial cleavage. We know cleavage is cell division without cell growth. Well, we're going to cover both of these things. The next slide talks about that fate of the blastopore. So we'll look at that real quick, and then I'll jump to the types of cleavage. So fate of blastopore is different between the deuterostomes versus the proterostomes. We said in this first picture, because these two things look identical. You've got a digestive tube, you've got 3 germ layers. The difference is that very first opening, that blastopore, in proterostomes, that blastopore develops into the mouth. Whereas in deuterostomes, that blastopore is going to develop into the anus. Small, minor difference there, but fun fact for the day. Here's the cleavage difference. We said cleavage, we've got cells dividing, but they have not yet started to differentiate. So that's cleavage. The spiral cleavage versus the radial cleavage, it has to do with the way that these cell layers line up. Look at the margins of these cells in spiral cleavage. You can see how this top layer of cells the cell margins don't line up with the second layer of cells. So if I were to take a knife and just kind of cut through those two cells to break this top group of cells into 2 pockets of cells, I would have to cut right through the middle of that second layer of cells. I would kill that cell by cutting right through it. So spiraled cleavage, where the cells don't line up, this is considered determinate, meaning that this embryo, let me do it this way. Before I say determinate, let me point out radial cleavage. Radial cleavage, you can see the cell margins from the top layer of cells and the bottom layer of cells. These actually do line up. The margins of the cells line up nicely. So if I were to take some type of laser knife or something, I could actually kind of separate the bottom layer of cells and the top layer of cells without cutting right through the middle of the cytoplasm of the cell. You could take this ball of radial cells and split them into 2 pockets of identical cells. That's why the radial form of cleavage is said to be indeterminate, because these groups of cells can spontaneously break apart. This is how you get identical twins. It's from one fertilized zygote that starts to grow into an embryo. And you've got a group of identical cells that's still in the true stem cell phase. But if it's an indeterminate type of cleavage, it's possible for these cells, the bottom and the top layer of cells, just to get separated from one another. And the cells can survive because you haven't split the cytoplasm of any cell, which is what would happen if you were to try and split one of these embryos from spiral cleavage. So spiral cleavage is said to be determined. There's not really a chance of this splitting and surviving, whereas radial cleavage is said to be indeterminate because it's not yet determined whether that's going to split into multiple embryos or not. So there's our distinction between spiral cleavage that's determinate versus radial cleavage that's indeterminate.
Continuous cell divisions in the zygote result in a hollow ball of cells called a blastula. With each cell division, the cells become smaller. As the embryo continues to grow, some of the cells of the blastula fold inward, forming the gastrula. All animal embryos, except sponges, pass through this gastrula stage. This opening becomes the mouth in protostomes, such as worms and insects, or the anus in deuterostomes, such as birds and mammals. The third layer of cells, called the mesoderm, develops between the endoderm and ectoderm of the gastrula. In deuterostomes, the mesoderm is formed from clumps of cells that break off from the endoderm. The mesoderm gives rise to muscles, reproductive organs, and circulatory vessels.
Yep. So we need to know those organs that are derived from any of those germ layers. I just like that little short animation because it shows that cell division and the cells are not growing at all. So that's our cleavage. And we left off right there where we were talking about different types of cleavage. So the next slide actually walks us into our second PowerPoint about animal diversity. And I'm going to once again turn the lights on real quick so that we can see the stuff that we've written on the board. And it's eerily similar to this slide that we have above us. In fact, the only thing that I've added here, if I can jump over to this side, it's given us the clade names kind of vertically here. This term metazoa just describes all of the animals. So it would include any of these animals, even the ones that lack tissues altogether. So the sponges are included in this group of metazoa. But you can see if we want to be a little more selective and just talk about true animals, the ones that have true body tissues, then we have to leave off the sponges and we narrow our focus to just the true animals that have body tissues. That still leads us to the separation between 2 layers of body tissues or 3 layers of body tissues. So we've got some examples of organisms with just 2 layers of tissues are comb jellies, the Ctenophora, and regular jellyfish, the Cnidarians. Then we also have our examples of triploblastic organisms that are bilaterally symmetrical. We'll see the flatworms or these acoelomates. And then we've gone ahead and introduced the ones that have some type of body cavity. We're going to see deuterostomes from this formerly known group as proterostomes, which has now been split into the term Lophotrochozoan that we need to explain a little further, and Ecdysozoan, which is where we're going in this next PowerPoint. Am I doing okay so far to this point? Haven't lost anybody. All right, let's switch to the second PowerPoint. What this slide is showing us is the sister taxa to animals, and it's what I've added up here to the board. I wanted to add an outgroup basically to our animals, and that's one of these groups of organisms that's not an animal, but it's a close living relative to animal, and that's these choanoflagellates. You can see a picture of one here. This choanoflagellate is a colonial protist. You can see that it's made up of a bunch of identical cells that are called collar cells. And you can see they have a flagella that they use to stir up water, and as water gets stirred up, any food particles that are on that water, any food particles that are in the water column, they get trapped along these microvilli. And that's how these cells feed. They basically filter food that's in the water column, and this flagella is what circulates that water to move across these little microvilli. So there's a protist that's made just out of these individual cells entirely. It's the choanoflagellate. That's why we consider it to be colonial, because there is no cell differentiation. There's multiple cells there, but they're all doing the same thing. So we said it's colonial and not multicellular. Whereas the most primitive animal here, the sponges, our group Porifera, they lack true tissues, but they are multicellular. That tells us that there is more than one cell there, and each cell is doing a different task. I know that we can't zoom in very much to this sponge picture right now, but tell me, from the very back of the room, can you see that it's like a light blue dot that we see in the walls of that sponge? when we get into invertebrates next time, we're going to look at that light blue staining cell that you can barely make out in the walls of the sponge. That's the other type of cell that make up cells of a sponge. There is this collar cell that is eerily similar to the choanoflagellite cell. It functions the same way with the flagella that stirs up water. Any food particles in the water column get stuck to the microvilli, and that's how this cell feeds. But in sponges, in addition to this collar cell, there are those little blue cells that I was talking about that are called amoebocytes. And what those amoebocytes do, they don't have a collar, they don't have a flagella that can capture food. Instead, the collar cell captures enough food for this cell and those amoebocytes. The collar cell, once it picks up food particles, they are packaged up in little vessels, they travel through this cell, and then like a baton race, this cell will pass off a packet of food to those amoebocytes. And it's the job of the amoebocyte to kind of run that food up and down the sponge to wherever it's needed. So sponges, even though they lack true tissues, they are multicellular. They got different cell types doing different things. Again, no tissue layers, just different cell types. When we get to the other branch, that's going to be either 2 germ layers or 3 germ layers, radial symmetry or bilateral symmetry. No coelom or some type of true coelom. Okay. This just shows us again that animals were around before the Cambrian explosion. This is a fossilized embryo that dates all the way back to 575 million years ago. So animals were around, they just rarely left any marks in the rocks. Here's one. This is one of those Ediacaran fauna. I'm going to kill the light again, and even though we don't know what this organism is, if I were to present animals that you've never seen before on the exam, you should be able to tell whether it is diploblastic or triploblastic just by telling the type of symmetry. So this organism here, what kind of symmetry would you say that it has? Radial symmetry. So that tells us it's got 2 germ layers rather than 3. It's diploblastic. What about this organism here? You can see a lot of segments. There's a narrowed end down here, and this is more of an expanded end. What kind of symmetry is this? Bilateral symmetry. And in organisms that are bilaterally symmetrical, we can see this cephalization. We see one end starts to congregate all the nerve endings to the head region. Once again, bilateral symmetry. And I mentioned that this Cambrian explosion was not really an explosion of, it's not like animals just showed up on the scene out of nowhere. They were around before the Cambrian explosion. They just didn't have hard parts. So we can really think of this Cambrian explosion as an explosion in animals that had an exoskeleton, just the development of an exoskeleton. It gave organisms that had it a huge advantage because they were harder to become prey items. It was a coat of armor that they're wearing. So what we can see in all these different animal body plans is bilateral symmetry, so we know we're looking at triploblastic organisms. The last couple of slides here just point out some of those differences, how the last bit of triploblastic organisms, they have become modified where that term proterostome is no longer used, and we now are going to introduce these terms ecdysozoa and lophotrochozoan. This is the old phylogenetic tree. And in this bilateral group, like we said, historically it was split into deuterostomes and proterostomes. And proterostomes were quite a large group. That has been revised. Proterostomes have been done away with. And this group has now been split into, here's the modern depiction of that tree. It takes into account that DNA and molecular data. We use the terms like I've put in here, parazoa, for the animal groups that lack tissues altogether, or eumetazoa for the animal groups that actually have tissues. And then if you have tissues, like we said, we've split it into 2 layers of tissue, radial symmetry, or 3 layers of tissue, bilateral symmetry. And deuterostomes have remained, but the proterostomes have been split into these terms, lophotrochozoa and ecdysozoa. Both of these are dealing with invertebrates. So here's this list. You can see that Proterostomes has now been done away with, and we've got our terms Lophotrochozoa and Ecdysozoa on here. This is a brachiopod. It belongs to this group right here, brachiopods. If you haven't seen them, they're kind of like a mollusk. They've got a shell, so they kind of look like a clam that opens up. And if we look inside of them, they are feeding with this structure called a lophophore. Here is that lophophore. It's a horseshoe-shaped, ciliated feeding apparatus. So it's using the cilia there to catch food items that are suspended in the water column, and it's pulling those food items into its digestive tract, either side of this little horseshoe shape. So this horseshoe-shaped, ciliated feeding apparatus is called a lophophore. Any organisms that have it in their adult stage or even in their larval stage are considered a Lophotrochozoan. So this is a lophophore in its adult stage, used as a feeding structure. If this lophophore is only present in the larval stage, it looks something like this. This is what we call a trochophore larvae. There's the mouth. It's still covered with this cilia feeding apparatus. In this case, only present in the larval stage. There's that horseshoe-shaped ciliated feeding apparatus that we call a lophophore. So Lophotrochozoans are feeding with this lophophore, and then the characteristic for the Ecdysozoans is just that they have some type of exoskeleton that has to be shed or molted in order for these organisms to grow. So if you take like a lobster or a crab, for example, they've got that tough exoskeleton. They're very protected down there in the ocean, but they can't grow unless they shed that exoskeleton. And when they do that, temporarily for about a week or so, they're soft. They're vulnerable to predators. And even this cicada is vulnerable to predation right when it breaks out of one of these, its first little, when it goes through its first molt, sheds its exoskeleton. It develops the wings. You will see this might be another good example of that morphogenesis that I talked about, where the larval stage doesn't have wings. It has a very well-developed digestive tract for eating food and storing nutrients. But when it goes through its first molt, it now changes where its digestive system shrivels up and goes away. The adult version here isn't going to eat any food. It's basically going to survive on the nutrient reserves that it's built up from its larval stage. It might live a week, it might live 2 weeks, and it's going to starve to death and die. Its only job in the adult stage isn't to eat, it's to find a mate and to reproduce. So we see wings and we see reproductive organs that mature. At the same time, the digestive organs shrivel and go away. So you have this real division of labor in the life cycle, where in the early part of the life cycle, it's all eat and grow as much as you can, and in the adult stage, it's reproduce and disperse offspring. So having to shed this exoskeleton or molt is called ecdysis. So Ecdysozoa is the name of the group that describes organisms that have an exoskeleton. So any of those exoskeleton organisms all belong to this Ecdysozoan group. These are two of the largest groups. I would say if we take 100% of animal species, 75% of those animal species belong to this arthropod group. I said something last time. I said that over 90% of the animals are invertebrates, meaning animals that don't have any skeleton backbone. Animals that we're going to consider vertebrates, like us, that have a backbone, they're going to belong to this phyla Chordata. I'll say it this way. If you're a vertebrate that has a backbone, you belong to this phyla Chordata. But if you're in this phyla Chordata, it doesn't guarantee that you have a skeleton backbone. There are invertebrates that belong to this Chordata phyla. Some of the more primitive chordates, like a hagfish that we talked about last time, it doesn't have a bony vertebrae like we do. Instead, it has a little ribbon of cartilage that I described. There is a blue planet one time that has a whale carcass. I'll try and find this video too, where that whale carcass sinks to the bottom of the ocean. Has anybody seen that, where there's just the hagfish that are tearing and ripping it apart? Those things can tie themselves in a knot because they don't have a bony vertebrae. They have a little rod of cartilage, so they're like a ribbon. We would say that doesn't have a true vertebrae, but it's in this group of Chordata. So some of the more derived chordates have bony backbones, the actual vertebrae. But every time I ask, list the 10 animals that are off the top of your head, we come off with like 9 out of 10 of them being the vertebrates. But that makes sense because that's the phyla that we belong to, so maybe we're biased towards that phyla. But there's a bunch of other different types of phyla that we're going to spend the rest of the weeks actually going through and introducing. So I think I only have in fact, that's only one more slide that I have left, because I want to at least set up how we start to break apart these different animal groups. And we have kind of set currently to look at poriferant. And to show you where we're going, we're going to see, I'm going to figure out how to draw this another way next time so that we can add detail to it. We're going to look at 2 different examples of porifera, 2 different sponges, a glass sponge and a calcium sponge. We're going to look at 4 different examples, 4 classes of cnidarians, and we're going to see 3 classes of platyhelminthes. We'll see examples of all of those in lab, and then we'll hold off looking at any other bilaterally symmetrical organisms until obviously a week from today. So just to preview where we're going in lab, we'll see examples of animals that don't have any symmetry and don't have any tissues. We'll see examples of animals that have radial symmetry and 2 layers of tissue, and animals that have 3 germ layers and bilateral symmetry. So it'll be a nice preview of all the different body plans, but there will be much more detail that we get into in terms of other things that those body plans can do.